Yes, ma'am. Yes, ma'am. You have to just simply present yourself, and after your when you finish your presentation, then the participant will interact with you, and that's it. You have to.
Now being recorded. Okay, 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 ma'am. Okay, ma'am.
Yeah. Yes, ma'am. So okay, ma'am. I'm leaving now.
Okay, okay, ma'am. Okay, ma'am.
Hello. Good afternoon, ma'am.
Good afternoon, sir.
How are you?
I'm fine, sir. What about you, sir? How it's going? This the program?
Yes, yes.
I think
Program is really
everything
going
going.
great.
Okay,
Thank you,
sir.
ma'am. Thank you for joining. If you could go to
Thanks.
the presentation and share your screen.
I have shared the screen. Already, ma'am asked me
Yeah.
to share the screen.
Sure, sure. Can you go to the presentation?
Yeah, yes. Shall I start, sir?
You can go to
But
the
but
presentation.
others have to join.
Yes, yes. We'll message them. If you could keep it on the presentation itself. Can you
Okay,
keep
okay,
the
sir.
presentation?
Okay,
You are audible as well as visible. Just the
okay,
presentation needs to be now visible. You need to click on presentation and start the slideshow.
okay. I have given. It is connected already.
Can you click on the presentation at the bottom?
Okay, okay, sir. Okay, sir. It's okay.
It's showing your screen. Yes. Can you click on hide at the
What
bottom?
about that next slide is getting?
No, yes, 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 yes. Can you click on first the hide at the bottom of your screen? Hide.
Yeah, hide. Actually, I can give it.
Yes, hide option, stop sharing and hide. Can you click on the hide? Do you see the hide option? Because this
Yeah.
is overlapping on the screen. Can you click on hide? Yes, 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 hide. Perfect. Now let's keep it as is. Let the team members join and we'll introduce you. Then you can start.
Okay, sir. Okay, sir.
We're sharing the link with participants. Let's wait few minutes for the participants to join. I think we should get started. Ma'am, if you could, Sonia ma'am, if you could introduce our esteemed guest. Dr. Sonia. Ma'am, if you could share your presentation so that we could start with the introduction.
Okay, sir. Okay, sir.
Ma'am, could you please click on the presentation at the bottom? PPT.
I have linked already.
No, you are sharing your screen, you are sharing your Meets, Google Chrome. You need to click on the PowerPoint at the bottom.
Yeah.
Yes. Can you start the slideshow? Perfect. Let me introduce. Good afternoon, everyone. Today we have the pleasure of having Dr. Jacqueline Pereira with us. Ma'am is Assistant Professor, Department of Aquaculture. Ma'am, if you could keep the presentation on.
Okay, sir.
Ma'am is Assistant Professor, Department of Aquaculture, Fisheries College and Research Institute, Thutkudi. Ma'am has a plethora of experience. Ma'am has a teaching and research years of 25 years. Her research articles, where she is the first author, is around 10 or more, and where she has co-authored, it's 15. And she has worked on 17 DNA sequences as first author, 17 DNA sequences as co-author, and she has served as co-PI in 2 projects. Ma'am, if you could start with your presentation, you're audible as well as visible, and your presentation is also visible. So let's keep everything as is. So continue with the presentation, ma'am, please.
Okay, sir. Okay, sir. Thank you, sir, for giving opportunity. Good morning, everyone. My topic today I'm going to share, that's molecular techniques in diagnosis of microorganisms. There are different diagnostic tests in microbiology. Actually, as you know, microorganisms, it is not visible to see our naked eye. So various techniques, with various techniques, we can able to detect and determine the particular species. So there are different ways, that's microbiological, biochemical, immunological, and molecular biological techniques. This slide shows the microorganisms. So one of the microorganisms given, that is with morphology and staining properties and growth on different laboratory conditions and phenotyping of the different protocol will give the microbe for specifically. First, we will with the microbiological techniques. First, we will go for a gram staining. It is I have given both gram positive staining and gram negative. For the gram staining, first we will give the for primary stain, that's crystal violet. Then with iodine, and then acetone, that's a decolorizing agent. then counter strain. If it is gram positive, it will be in the purple color, and if it's a gram negative, it will take only counter strain, that's saffron. If it is gram positive, we'll go through the following list of tests I have given. And if it is gram negative, these are the tests will given to identify the gram negative bacteria. It will give positive result in these tests. This is example, my mannitol salts test. Agar, the mannitol salt agar, if it is yellow color colony, there is a positive. If it is pink as like that of the media, there is no change, that indicates negative. Then in the glucose broth with the Durham's tube, that is for positive with yellow color, pink with negative. And blood agar, here the clearing zone in the blood agar, that indicates positive, and otherwise it is negative. Then methyl red and bogus process, the two reactions has given here. That is positive for purple color and negative for the yellow color. Then cement citrate, that is green colors, that is for the positive, and citrate for the that blue color, that is for the negative. And coming to the immunological assays, here the antigen antibody reaction, that is tested. That is if it is during the immunoassays. The ELISA is one of the immunoassay. Like this, Western blot can also be tested. Now we'll see our molecular techniques. How to actually molecular techniques? It's nothing but it's actually we are going to test the molecules that is DNA level, or that is in the that is present inside the nucleus. That's why this molecular level, the DNA, mitochondria, cell membrane, cytosol, all these components present inside the cell. So what you are going to deal with the DNA that is inside the nucleus, it is present. So what is molecular diagnosis? If you are test with the DNA, RNA, proteins to test the specific genes. So why molecular test required? It is required because it give accurate and sensitive and timely diagnosis within 4 hours period. It is able to diagnose. During the corona outbreak, it is RT-PCR able to give the result within 4 hours. So this such a short period we can able to confirm. The previous case we have discussed the molecular microbial techniques and all. Microbial techniques, it will take at least 2 weeks of 15 days period to confirm the entire analysis. If you want to go for immunoassay is also it will give after 15 days only we can collect anti-serum for the testing. So it will take also 15 days period. So that much. So with this molecular technique, within the day itself, we will give the we can able to specifically identify the particular pathogen. So it is give the proper measures, and it will give the wastage of time, energy, money towards that. So let us see the area of molecular diagnosis. It is not only in our microbiology, it is applied in various area like species identification of any species, any plant, any animal, whatever that is. The population studies, it will able to compare the wild and natural population. It is the demographic, the particular species is having bottlenecks in the particular area. So that things can be able to read. It is applied in rehabilitation program. This is diagnosis. That is my microbiology. For the pathogen identification. Then for food fraudulence also it is applied. It is applied in drug metabolism and analyzing best, and it is so this is a short period since we are analyzing with molecular techniques, it is able to give best and appropriate therapy. So let us see our molecular biology. This is the picture shows the DNA. DNA is a negatively charged one. So it will here the hydrogen bonds are given. It's a double helical structure with the hydrogen bonds. It is binding together. The two single strand bound together with the hydrogen bonds. As you know, that the scientists Watson and Crick, they are the Watson and Crick. They are the discovery. They Rosalind Franklin was information that discovery used by Watson and Crick to develop the double helical model, and they got a Nobel Prize for that one. So let us see one by one various techniques. First, we will discuss about the gel electrophoresis. What is gel electrophoresis? Here, the already seen that our DNA is negatively charged particle, so it will move towards the positive electrode. So here under electricity, it is getting the molecules get separated. So that is what happening during gel electrophoresis. It was first observed by the scientist during the year 1807. Here this will by this method the molecules get separated because of the charges of the biomolecule, and under electricity various the electricity, if you increase it is 100 volt, it will go faster. If you decrease 80 volt, it will decrease like that, based on the electricity and matrix, which matrix you are using, agarose or polyacrylamide gel, like that, and buffer, which buffer you are using, TAE buffer, TB buffer, tris acetic acid, EDTA buffer is TAE buffer, or tris boric acid, TB buffer, like that buffer solution which one we are using. Here this picture shows how we have to add DNA samples into the electrophoresis apparatus into the gel loading gel. So this is one of the picture of a PCR product under UV light, how it will it is visible. So this documentation with the gel electrophoresis. So the coming to the again, this electrophoresis, what are the factors affecting the mobility of the molecule? First with the molecular factors, that is charge, size, shape. Then environmental factors like electric field, strength, the matrix, running buffer. This is the electrophoresis apparatus. Again shows in the vertical position. And let us see that what are the types of matrix paper, nitrocellulose paper or nylon membrane can be used, or agarose. It can be polyacrylamide gel or agarose electrophoresis. Based on the pore size, that's based on the concentration. If it is 1%, the size of the pore will be high, then it is 2%. So the molecules will be, the concentration high means molecules will be very closer. So pore size will be less. So then acrylamide gel like this, that is also odorless crystalline solid chemical. It is again also like this agarose, it can use. This will you can be able to soluble in water or ethanol or ether or chloroform. And this here acrylamide, that the fine particles, smaller particles able to separate with this acrylamide gel. Then starch or cellulose acetate can also be used as a medium. Then in this agarose electrophoresis, you can see the DNA band pattern, how it will be, how you can visualize with fluorescent dyes with the ethidium, like ethidium bromide. You can use as a fluorescent dye to determine the molecules, because it is act as an intercalating agent. Then coming to the blotting, once the DNA is in the gel, from the gel to the blotting, like if it is DNA present in the gel, that can be blotted with in the nylon membrane or nitrocellulose membrane. So such kind of blotting, we can call it as southern blot, because the scientists, those who did the southern is the name of the scientist. He did that one, that is given to honor his name, he has given as a southern blot. If it is that is given for DNA, once southern has given for DNA, and then for RNA, northern has given. For the protein, western the term has given. In the southern blotting, there are two important features for the hybridization. This is the reactions are specific. Probes are applied. Probes are nothing but single-stranded nucleic acids. That will probe means it will seek where exactly complementary sequence is there. So probes are single-stranded short sequence of nucleic acids. So it will seek where exactly complementary sequence are present in our template or our particular gene of interest, which one we want to detect. So in the sudden blotting, first so the DNA, if after extraction of the total DNA, we have to go for the cutting with the restriction enzyme. These restriction enzymes are nothing but they are the biological scissors. It is extracted from the particular bacteria. So based on that, we have to give nomenclature for the restriction enzymes. So yeah, actually, the restriction enzymes are nothing but biological scissors. It will cut at the specific site of the particular DNA. So that will, if I add DNA with a particular restriction, a specific restriction enzyme, it will cut at specific sequence of the particular DNA, and we will get as a different fragments of DNA. So the such fragments of DNA will be denatured. So the way we have to go for denaturation, we should get single strand. Then only our probe will go and attach. So for that, it should be denatured, and the probe will be hybridized. Hybridized or bind with the complementary sequence where exactly. So then it should be. We have to use some labeling thing like a fluorometer. If you are using fluorescent dyes, or if you are using radioactive compounds, radioactive isotopes, if you are using that radio detecting instrument is autoradiography, like that will occur. Then, like this, if it is RNA as a starting material, we have to go for northern blotting. Instead of DNA, here it's RNA. If any gene expression studies, so mRNA, if we want to test, means we can go for RNA blotting. So the same way was everything is same, only the starting materials RNA. Then coming to the protein, if it is a Western blot, means it's a protein. Instead of the sample DNA or RNA, here protein. So protein will everything is same. So but here we need STS page. Page is a polyacrylamide gel. STS is nothing but sodium dodecyl sulfate. So the STS is nothing but it's a sodium dodecyl or sodium lauryl sulfate also, you can say, that will denature the protein. So that will help to bind our probe. It's a specific antibody will be we can use for the detection of the particular known protein or unknown sample containing protein. So once we are using, and that will from the gel, it will be transferred into the membrane, nitrocellular membrane, nylon membrane. Then we will be adding that probe for the detection. Detection of probes, already I mentioned, it is probes are nothing but single-stranded nucleic acids. If in the case of a DNA or RNA, Western blot or the southern blot, the southern blot or northern blot. If it is a protein, means Western blot. So it should be labeled radioactively. If it is, we have autoradiography, we can label with P32, iodine on like that. Radioactive compounds, isotopes can be used for labeling. Otherwise, we can use fluorescent dyes. Fluorescent dyes can be used, or enzymatic labeling also. So fluorescent dyes, like kindly mute. Please kindly mute somebody's background sound is getting. So sir, ask them to mute. So some background noise is getting. Sir, what to do? Shall I continue, sir? Sir, shall I continue? Okay, some background noise is getting, that's why. So the probes, so for the detection of probes, radioactivity or fluorescent dyes can, or even enzymes like the radish peroxidase or alkaline phosphatase can be applied. So this is one study detected that the X-ray film or autoradiography can be used, or a fluorescent dye spin fluorometer or fluorescent microscope, like that. You can use the enzymatic activities, like if you want that HRP, horse peroxidase or alkaline phosphatase can be used. Let us see the molecular techniques. Next techniques, that is PCR, polymerase chain reaction. This is here what is happening. The technique able polymerase chain reaction in the technique in which it is able to generate the highly identical copies of a number of copies. So it is possible with the cloning technique as well as PCR technique also. Here, this technology was discovered by the scientist Carey Mullis during the year 1983, and for his work, he is a biochemist, and for his work he got Nobel Prizes during the year 1993. And this technique in which the thermal cycler DNA engine is applied. And the Carey Mullis, he shared Nobel Prize with his colleagues, Michael Smith and Mollis. This picture shows polymerase 10. So here you can see the picture. This, the two strands is getting during the stage of denaturation. It is separating. Is double strand into two single strands. Then in the annealing step, that is the primer. Primer is nothing but it's a new single stranded nucleic acid. It is primary specific part for a particular gene. It will bind where exactly the complementary sequence is available. Here for this reaction will be carried out by the enzyme, that is polymerase enzyme, DNA polymerase enzyme. Actually, enzymes are nothing but they are the proteins. At high temperature, it may get denatured. So to avoid the situation, the enzyme, that DNA polymerase is the enzyme. It is more apt. It is not getting. It is stable even at very high temperature. So at 95 degrees centigrade, also it will not happen. It can able to withstand. It was extracted from the bacterium Thermus aquaticus. It was from the hot spring. And so if we are giving, so that will polymerase DNA and by adding DNDPs. And we give billions of copies.
I want to read for Bing.com.
So it is able to give at the end of first cycle we will be getting 2 double strands. Like that, the second cycle 4 double strands. So like that 8, keeping on multiplying 18, 8, 16, 32, 64. Like that 2 power n. If the number of cycle is 35, means 2 power 35 copies will be obtaining. So this step again, you can see this picture also, where we can go for a documentation. You'll get the gel picture like that. The step 1, what is denaturation? It's separation of the strand is at high temperature. So and in the PCR master mixer, we will be adding magnesium ions also. That will stabilize DNA, and it will serve as a cofactor. During denaturation, the DNA double strand get single strand. And the priming or annealing, it's reducing temperature from 95 to or 94 to 15 to 25 degrees. That is based on the which gene, based on our desired gene, we can have the annealing temperature. And if the temperature increases, specificity increases, but the yield decreases, or reverse versa. Then the third step is polymerization or extension. Here at the end, so this step will be carried out by the DNA polymerase enzyme, that is a TAC DNA polymerase we are using. Approximately 150 nucleotide per second is adding. So how this PCR works? So PCR actually it is containing its synthetic oligonucleotides we are adding in the PCR master mixer, and at high temperature, that is it will denature at 94-95 degrees centigrade, and rapidly cooling, that's annealing, that's get reducing. Those primer will prime, initiate the DNA synthesis, and extension that is well with our thermos aquaticus will extend at the temperature usually 72 degrees centigrade is giving. So it will extend. At the end, we will get at the end of the first cycle, we will get 2 strands like that. After the first cycle over, again go to the denaturation, then annealing. Like that, the cycles will continue for 35 means 35 cycles, or 30 means 30 cycles. So at the end, there will be a final extension, that is again the temperature 72 or 70 degrees, 70 or 75, 70 to 75 degree can be given. Then once that is given for 10 minutes can be given. After that, if you are the scientist, those who are working, in particular, he is doing some other urgent work, means he can keep it for for the 4 degree forever. So when he will find when he is come till his arrival, it will remain as 4 degree without any problem. To the PCR products is safe. Quality is good. So this is this picture shows the thermal cycler. We can give programs like that, where if it is here, it has given initial temperature, initial denaturation 7 minutes at 94 degrees centigrade, and in this step 2, that is 45 cycles, has given 20 seconds 94 degree denature, and annealing 64 degree, extension 72 degree. This is the first cycle. Then again the first second step, continuously once extension over, again move to the denaturation, annealing, extension, like that. 45 cycles will complete. Then again final extension 7 minutes, 72 degrees centigrade. After 7 minutes, it will hold for 4 degrees centigrade as a storage for a forever. Like that, we can give the program into the DNA engine or DNA thermal cycler. Let us see what are the optimizing factors actually that is starting the nucleic acid, DNA, RNA, and the TAC DNA polymerase have to use here. Otherwise, other DNA polymerases will denature, and we have to use the specific primers. The primer we have to design in such a way that we should get the particular gene. We have to give the particular its primary specific for a particular gene. And already I told the PCR mixer containing magnesium ions and TRIS HCl and DNDPs also. DNDP is nothing but a nucleotide for triphosphate. That is a adenine triphosphate, cytosine triphosphate, guanine triphosphate, thymine triphosphate, like that. How this PCR is powerful. PCR can amplify usable DNA. Actually, where the nanogram, it is available only the sample is nanogram. But this powerful technology, we can amplify into millions of copies. So it can able to detect. Otherwise, if you go for microbiology, immunology, and all, you need a sufficient quantity of the sample, then only will give the result. But here it is not. If any disease in the initial phase, we cannot get the sufficient quantity of the sample. The pathogen level will be less during the initial level. So that time also it is possible with the PCR technology, because it will multiply into millions of copies. So it is able to give the specific pathogen which one is actually affecting, like that we can be identified. So this technology, it is the enzymes take DNA polymerase. So by this technology, we can use even any digestive enzymes can be used, or even the single sperm is able to give the answer. The father may deny. So patent testing, it is possible with a single sperm like that. So this applied in our clinical microbiology, it is even the small any disease. That is why during the the pandemic situation, the nasal cavity, because asymptomatic person also it is able to give through the, if the person healthy means there is no clinical symptoms. So vocal cavity sample or nasal cavity sample during the entry itself, entry of pathogen through the nasal cavity or vocal cavity, that can also be tested with this PCR technology. So rapid detection is possible with PCR techniques. Then let us see the other molecular techniques like RIPD. Here in the PCR we are using specific primers. Here RIPD, this is not so for PCR we must know the knowledge of the primer sequences. But in the case of RIPD, without knowing the primer, random primers we can use without knowledge of the primer, without the knowledge of the particular genes. We can apply random primer for this for our identification part. So that is why it is randomly amplified polymorphic DNA. Everything is same. Instead of specific primer, we are using random primer. So first here also the steps are same. Isolation of DNA, that's for any molecular study, and purification of DNA, then amplification DNA already we have studied in I have discussed it in detail. Then once it is amplified, we have to go for gel electrophoresis. This picture shows what is happening in the isolation of DNA, then purification. With a kit, you can purify, and electrophoresis apparatus for the separation of DNA. We can separation of DNA, then you can documentation, gel documentation. Then the next techniques is RFLP, restriction fragment linked to polymorphism. Here here also the first step for any molecular study, first step is DNA extraction or DNA isolation. So that should be that the extracted DNA should be treated with the restriction enzymes. Already I have mentioned in the introductory part that the restriction enzymes are nothing but it's a biological scissors. Biological scissors, it will cut at the specific site. So we will be getting different fragments from the total DNA. We will get different fragments. So during electrophoresis, we will get different fragments in the gel. Once we get the gel with the different fragments, that can be transferred in the blood into the different membrane like nylon membrane or nitrocellulose membrane. Then it should be transferred into the X-ray film. Then we have to add a radioactive labeled probe, and that will be detected with the under autoradiography. That is RFLP. This here actually we will get the PCR RFLP pattern like that. Then coming into the next molecular techniques, that is amplified fragment length polymorphism. This is digester here also. Everything same here also, no need of the prior knowledge of the particular genes. So like RAPD here, this is also without knowing that primer, we are using some enzymes. First step is genomic DNA extraction, then followed by restriction digestion, and then we will be using the adapter molecules. But here we are using restriction digestion, not any enzyme, some specific enzyme, EcoR1, MSA. So which enzyme we know, if it is using EcoR1, we will get DNA sequence like that. So that's why we are using the specific restriction enzyme. Once we get the different fragments, that should be linked with the adapter molecule, ligate with linking with adapter molecules. That also nucleic acids. So once linked with adapter molecules, then regular our PCR steps, denaturation, annealing, extension, like that. So we can able to multiply and we can go for electrophoresis. Once amplification over, mean electrophoresis, the particular polyacrylamide gel, the gene expression studies, small molecules can also be identified with acrylamide gel. This AFLB bands show, this picture shows the AFLB bands. The next technique is single nucleotide polymorphism. This here, if a single gene get mutated, that can also be tested by this method. Here, instead of GC, if it shows AT, GC means guanine and cytosine. It may be normal one. If it is get mutated, instead of cytosine replaced by thymine or guanine replaced by adenine, means that can also be detected. A single nucleotide of the particular gene, the functional gene, have a single nucleotide get mutated, means that leads to drastic changes in the body. So that can be detected. And then coming to the next technique, that's DNA sequencing. Sequencing is nothing but in which order the nucleotide is arranged. The previous one technique, single nucleotide, you think a particular nucleotide changes can be tested. So here, what is the exact order of present in the particular genome, that can be tested with the sequencing method. There are two methods are there, maximum Gilbert and the Sangus dioxy method. So what is the maximum Gilbert method? This is a chemical degradation method. Here the purines damaged by dimethyl sulfate group or a methylation of the base or heat releases at high temperature. Also in the PCR and we have seen at high temperature single strand get denatured double strand get denatured into single strand. So heat releases the base, so it is able to detect. So like that, alkali also you can use with the sodium hydroxide or different chemicals can be used as alkali, cleave the Ag molecule, like that. So this technology, first maximum Gilbert is chemical degradation method. Now we are in practice the method of DNA sequencing is a Sangus method. It's now we are practicing Sangus dideoxy method. First, actually, before sequencing Sanger DNA, he has sequenced the protein during the year 1958 for the protein. He's Frederick Sanger. His name is Frederick Sanger. This technology developed. He developed sequencing of amino acid first during the year 1958. Then later phase during the year 1980, he has developed dideoxy sequencing for the DNA. So what is happening here? What is the principle here? Here also the polymerase in reaction, we are using tag DNA polymerase, but here the DNA will be nitrogenous base as AGTC. That will be replaced by dideoxy nucleotide, D and DP, dideoxy nucleotide triphosphate. Then separation with electrophoresis, then we can read the DNA sequences. So here you can see the picture, the top you have given which sequence we want to sequence. A particular DNA fragment is given. For that we don't know what is the exactly its sequence, but here that the dideoxy for each nucleotide it will get terminated. So you can call it as chain termination method also. For each nucleotide it will get terminated. So at the end we will get the complementary sequence of that one. From that arrive what is exactly our sequence. So here that for the while going for electrophoresis, you will get the pattern like that for each nucleotide with a different color pattern. This actually initially manually did DNA sequencing. What is exactly happening here? The different fragments of DDD, dideoxy ATP, dideoxy CTP, dideoxy GTP, dideoxy GTP. So separately separated, and it will be given in the order in the gel during electrophoresis. Which one is first? That is moving. So it is based on the which one is first produced. So based on that, it will be given in the pattern. So from that, we can read the sequence of the particular DNA. When we compare with the two methods, Sanger's method and Maxon and Gilbert chemical degradation method. So Sanger's method is a enzymatic method. Yes, that's tag DNA polymerase using. It's for the chain termination. It's this one chemical methods. Here, the Sanger's method, because DNA synthesis here, it is here also it requires DNA. Here the chain termination method, here it is a long stretch of DNA get degraded. It will break into different fragments. So all these things we have studied the various techniques about the nuclear DNA. You can see inside the cell, molecular biology in the previous, initially I showed the slide with the cell inside nucleus, inside with the cell organelles, and the nuclear DNA present inside the nucleus. So this is the nuclear chromosomal DNA that is in the network, chromosome network form, it will present inside the nucleus. But apart from that DNA, nuclear DNA, there is mitochondrial DNA also present. So the mitochondrial DNA, there are 37 genes are there. It is essential for normal mitochondrial function. And this DNA, actually mitochondrial DNA, it's a maternal inheritance. There is no paternal inheritance. And this provides again instructions for the particular gene. So this is the structure of mitochondrial DNA. Actually, myself, I have worked in my PhD work on mitochondrial DNA of the particular at the end I'll explain that one. About the mitochondrial DNA somewhat elaborately. Then coming to the real-time PCR. Real-time PCR is nothing but here we are quantifying the DNA. What is quantifying DNA exactly? How much qualitatively we have tested the other techniques method. Here the real-time DNA, how much, how far, what is the intensity of the disease, or what is the expression of the particular gene intensity can be studied. So here the gene expression we will have RNA, mRNA, so that can be go one more step. It should be converted into cDNA, complementary DNA. So with before that, how RNA will be converted into DNA, we need one more step, that reverse transcription. For that, the DNA reverse transcriptase enzyme have to use for the purpose of converting RNA into DNA. So we will be getting complementary DNA. The complementary DNA, once we get the DNA, then the other steps are the like that PCR. So that's why it is real-time PCR. So let us see the choice of markers. How will you apply? The mode of the different markers, how will you select the rate and mode of evolution? You have to select mode of inheritance, mode of expression, availability of sample, sensitivity, availability of the marker, single locus or single nucleotide. We have discussed it, and relatively cost. Then coming to the advantages, it is highly sensitive. Molecular techniques are highly sensitive. It is have high specificity. It is with a faster manner you can able to ascertain the result. So speed is high, and simplicity, it is now automated. But coming to the disadvantages, it is highly somewhat expensive thermocycler and all record. And chemicals, molecular biology, chemicals also something there. And there is sometimes we miss some new organisms. Sometimes some false results, or if the person is not doing properly also some false. So again it is too sensitive, less clinical value. Even the less clinical value or sample also, it will give too sensitive. So thank you. This now you can, now it is now it is open. If any doubt, actually, for in my PhD work, I did DNA barcoding of